Okay, do you want me to stand over there so you can see if it looks okay? Or do you want
No, it's fine. I put there.
Yeah.
Okay. There. We're live. I put the Instagram too.
Next time I'm at UCLA, I'll see if I can find out what's going on with the coyote study.
Oh,
I've messaged a few times to find out if something's holding up, because I think we should be getting data back.
I think they sent some files, so a path to. I'm not sure. Thank you. I haven't checked that in a while. But yeah.
If my pictures aren't terrible, I can send them to you as well.
Right. Did you need this too?
Not now.
Okay.
Thank you. Yeah, I have the computer busy.
Oh, okay. All right. She's got copies of all the photos I've taken so
Oh,
far.
perfect. Thank you. Yeah.
Thank you for letting me borrow that.
You know, this was my idea because one day I was walking in a heat wave in New York, and I went into a CVS,
Mhm.
and I saw it. I was like, "Oh my god, that's so cool!" And I was like going around, and they were joking, like making jokes about me that I looked like an old lady with my thing. I'm like, "We should get it for us." I'm like, we ended up getting it.
Nice.
Ended up as a joke. Yeah, it started as a joke and ended up as a great idea for this one.
All right, good morning. My name is Sebastian Garcia. I'm going to be moderating the third session of the MetaSUB conference, the Two Frontiers project session. Just a quick announcement. At 10:15, we're going to be having a 15-minute break, and we're going to start with James' session at 10:30, starting from there. As a reminder, the talks are going to be 25 minutes and then 5 minutes for Q&A. In the interest of time, I'll make sure to keep us in that 5-minute range. I know it's a very interesting set of conversations, so, if the time runs out, I invite you to, continue talking afterwards. For the first session, we're gonna have Dr. Braden Tierney. Dr. Tierney completed his PhD in biological and biomedical sciences at Harvard Medical School. His current studies focus on the intersection of microbiology, bioengineering, and machine learning. Dr. Tierney is also the co-founder and executive director of a group called the Two Frontiers Project, which we'll hear about more today.
Thank you, Sebastian. Thank you, everybody. It is wonderful to be here talking to this group today in my capacity as the executive director of the Two Frontiers Project. And the purpose of this talk is to queue up the next session that you're going to hear and tell you a little bit about who we are, where we came from. We spun out from MetaSUB, that's why we're talking to you today, what we're doing right now, and where we're going. So I want to start with who we are. This is the core question that kind of motivates 2FP. This is what we think about. You probably know the movie Jurassic Park. famous line, Jeff Goldblum kind of murmurs, life finds a way. We are interested in how, especially when you don't expect it to. Like in humanity's greatest frontiers, the depths of the ocean, volcanoes, hydrothermal vents, the reaches of space, the International Space Station, all these places, these extreme environments where amazingly life figures out how to survive, and the mechanisms of survival are what intrigue us at the Two Frontiers Project. And so I like starting with a sort of sense of an acknowledgement slide, because this is a massive project. It's ballooned. We have so many collaborators, so many different people. You're going to hear from a number of them today. I hope we have future collaborators in this room. Very excited about that. But to really understand kind of how we got to this big list of people, it actually starts with this name up here in the corner, who is not here today. It starts with Ted Chaffigan. So I'm going to give you the kind of 2FP story. So in 2021, Ted sent me a Slack message, and he said, "This is cool." He sent this paper that was by a number of collaborators from the University of Palermo. One of them was Marco Milazzo. Ted and I we got our PhDs together. He's a brilliant bioengineer and microbiologist. And we were having a lot of conversations about the most interesting places in the world to sample. And so following that conversation, he sent me this paper. And I ended up actually getting in touch with the authors of this paper, specifically Marco Milazzo, saying, "Hey, has anyone ever gone and done MetaSUB style shotgun sequencing in these shallow CO2 seeps, these environments where they're really unique because they're volcanic seeps, where you have the ocean basically saturated with carbon dioxide in the photo zone, like in 10 feet of water. So you might find cyanobacteria that are really good at eating CO2. You might find all these interesting things." And so I asked Marco, and Marco is an amazing collaborator. You're going to be one of his former students, Davide, later today. And he introduced me to Paolo Baltrini, and I got Chris O'Ryan and Chris Mason involved in the MetaSUB connection. Let's write a grant. Let's write a grant to go and actually raise money to go sample these seeps and see if we can find microbes that are really good at eating the CO2 that's there. So we wrote this kind of wild title, A Tale of Two Frontiers. That's where the name comes from. Naturally evolved carbon dioxide remediation for Earth and space-based applications. And the premise is very simple. We're going to go to the seeps, we're going to sequence, we're going to culture microbes, and we're going to find the future of carbon capture. So we wrote that grant, we submitted it, and here was the general concept. These seeps are used to study ocean acidification. They could have new mechanisms for capturing carbon that would be relevant for wastewater treatment, CO2 sequestration, to novel bioreactors, potentially the metabolisms you would see there, you actually would expect to be relevant for cleaning up and bioremediating corals when they get contaminated with all sorts of stuff and runoff. There's all sorts of interesting thermodynamics going on in these seeps, and we wanted to sample it. We came up with this insane set of aims. We're going to do all of this remotely. We're going to build deployable kits. We're going to do all the microbiology on the island of Vulcano off the coast of Sicily. Then we're going to ship everything back, and we're going to do this culturing and paired sequencing in the States. It's going to be wild, and of course that grant got rejected because it sounds insane. And that does not exactly, that's not how academic funding necessarily works. However, if you know Chris, you know me a little bit, we thought we were going to try again. So we went and we got in touch with one of my best friends. I've known him for about 10 years. We worked together a decade ago at a company called AgBiome, James Hendrickson. You're going to soon find out that he is one of the most brilliant scientists and microbiologists you can ever find. And with James, we refined our proposal. We refined a proposal and said we're going to actually get some nice figures in there. We're going to design specific enrichment conditions on the island based on the geochemistry of the seeps. So identifying, for example, based on this paper, there's a lot of sulfur oxidation reactions. James was able to use this to design enrichment media that we could then inoculate on the island. You saw a wonderful talk yesterday about the importance of doing microbiology in the field to replicate the state of nature with your culture. So that is what we think a lot about. So P and James really brought that strength. Now, we wrote that grant, we submitted it again, and it got rejected again, because, again, it still sounded insane. So, in parallel, though, I had been working with Seed Labs. This is one of the CEOs of Seed, Raja Deer. And Seed is a company, they have a philanthropic arm that does a lot of interesting philanthropic donations to look at applications of biodiversity to solving major problems. And so I said, "Hey, Raja, are you interested in potentially donating some money to fund a tiny little expedition to Sicily to do this crazy thing?" And Raja said, "Sure." So we ended up putting together a bigger team. I reached out to some of my favorite microbiologists, again, Max Schubert, a guy I did my PhD with, to do some more culturing back to the States. Seeds and John Kowitz, you're going to hear from him later today, to take some beautiful photos. We was introduced to Gabriele and Davide, who were Marco's postdocs, and we put together this big team to go actually do this sampling. And we went, and lo and behold, after that one expedition, We actually, here we are diving into seeds. These are photos taken by John, sampling sediment, water, biomass. These are some of the cultures and enrichments that Max and then Ted did back in the States when we shipped enrichments back to them. And we ended up finding this one really remarkable strain, Cyanobacterium pepininum, or volcano, that grew very fast in CO2 and produced a lot of biomass. And that was really exciting. And we're going to talk more about that in a second. This is James. We built a remote lab in a hotel room. This is James, very sweaty, working in that hotel room. You can see the water sediment and biomass sitting there. You can see all the tools we shipped out. This is all very, on the fly, putting this together. We've since gotten a lot more refined in our techniques, and you're going to hear about that later. So these are some of the, we have one paper in press right now on this strain, this UTEX 3222. Basically, Max streaked out a bunch of stuff on different agar optimized for growing cyanobacteria. He found this strain that grew faster than UTEX 3154 and these other comparison strains. It was huge. The size of these cells are gargantuan compared to what you would normally expect. It was not phototoxic. It was planktonic. It was tolerant to all sorts of temperature, pH, and sodium, and different light ranges. And it produced a massive amount of biomass and compared to comparative strains. If you know anything about carbon capture, these are really good characteristics for a potentially bioproduction industrial development strain. Robust to all kinds of growth conditions, produces concentratable biomass quickly. And the coolest result from this was that it sinks very, very fast. So if you look in the two tubes on the left-hand side there, left one, UTEX 3154, comparative similar strain, a chassis used for microbial or carbon capture. And then on the right-hand side, you have our strain. They've been sitting in the test tube stationary for the same amount of time. Our strain sank to the bottom in 12 hours. Really cool phenotype, one I really want to explore. And then we in this paper go on and talk about how it can be used for concentrating biomass, capturing carbon, reducing tree mass to compare it to how many trees it would take versus a couple liters of this stuff. And this is all really exciting. Here's our preprint. I can share it with you afterwards. And we're not going to get into the whole carbon capture thing right now, because what this experiment showed, those funders, is that this concept works, right? Going to a place where there's interesting geochemistry, and then culturing things based on that geochemistry can find novel microbial physiologies that could be really relevant for all sorts of interesting societal problems. So, with that one result, that one incredibly laborious expedition, which happened because of Chris and so many of the people here, I'm now going to tell you what that turned into. So, fast forward a couple of years, we now have 3 active funded initiatives. We have, I don't know why it's like a focus on C's, we have our crop, spoil, and carbon initiative that kind of, limits what we can do, so we have to go like clinical or something next. But we've had multiple expeditions around the world, all these different continents. We have now a very large data set, which we'll talk about, of high CO2 seeps and terrestrial CO2 springs. We have a NASA-funded grant to go try and grow crops in lunar regolith based on microbes that are growing in the Mojave Desert, on plants in the Mojave Desert. We have a whole data set of corals that we're building from around the world, completing coral draft genomes, building methods for sequencing the coral biome to design precision probiotics to increase tolerance to global warming in the water. So all these different projects, citizen science programs, all sort of in the MetaSUB vein, inspired by what this group did, and it continues to do. And so this, you saw that slide, that little graphic that we put together on, water sediment, biomass, enrichments, and then application. Well, this is what it's evolved into. We now have this full pipeline for identifying interesting geochemistry in the world, having these deployable Pelican cases we can drop ship anywhere, design media on site, inoculate into that media, you're going to hear about that from James later, and then design downstream screens like sinking microbes for carbon capture that are very applied and focused. And of course, it's all backed up by a nice bioinformatic pipeline, which is, as you guys know, my thing. And so the reason why we can focus on all of this is because microbiology, microbes are ubiquitous. The machinery they use to survive in these environments is very different, but at the end of the day, they're self-assembling machines. So you can use a similar set of Pelican cases and tools to explore very diverse physiologies. You have massive economies of scale for microbiology. These are example kits that we have put together. So we have cap numbers and everything we can talk about. I'm really excited to collaborate with all you guys on the deployable science stuff, because it's something we've worked a lot on, so I really loved that talk yesterday. And so we basically try and standardize everything, everything can be shipped out. We've done a lot of work, and I'm reason to this because you're going to hear about in the different talks later today on optimizing enrichment conditions based on field measurements, everything from simple aquarium strips to evaluate the thermodynamic reactions occurring in a given environment. Krista has gotten really good at piloting shotgun genomics in the field. You've heard a few talks on that today. This is now running on the hood of a Honda pilot. in a snowstorm. And that was on our second carbon expedition in Colorado a couple of years ago. We've been using that information to also inform sequencing and cultural conditions in the field real time. This is a picture of actually an old version now of our deployable media kit. You can make about 5,000 combinatorial media on site from that Pelican case. We can do OPs on site and through simple 3D printed equipment. That, we actually have that here if anyone wants to see it, is about, what is it, $200 3D printed microscope. The most expensive part is the objective. You can do phase contrast, all this wonderful stuff, ship it around the world. You're going to see this image again. These are some of the different kinds of growth we get. We design enrichment media on site, very diverse physiologies, again, based on the geochemistry of what we're seeing there. Okay, so our carbon initiative, you can see we have these sort of 5 buckets. And the goal is to really understand the evolution of microbes in light of carbon dioxide at varying concentrations around the world. That's the sequencing aspect of it. From the culturing, of course, we want to develop new applications for carbon sequestration. We've gone to Volcano and Alien Islands now twice. Rocky Mountain Springs, very high CO2 springs out there. The one that we're announcing and talking about today is after this conference. We're actually going to Shikimajima off the coast of Tokyo, where they also have shallow marine seeps and corals exposed to those marine seeps that are tolerant to the acidic water, which is really interesting. So we're going to be sampling both carbon seeps and corals there, and doing all of our on-site enrichment, everything, all that work, and sequencing on that island. And so yeah, we're now on the order of hundreds of samples, thousands of enrichments, and actually we have thousands of isolates at this point. One other brief technical note, this is, so I want to emphasize, John Coates is going to give a talk next. And, you know, John is amazing to have along because he's an incredible photographer, because he also has an extraordinary eye, right? So John actually worked with us to spot this interesting red biofilm in Volcano. And he basically went down, it's a great for your eye, we went and grabbed a sample, we took it, we put it through our enrichment process, sent it to TED, University of Wisconsin, who put it through chemostat growth for about 2 weeks, and looked how the community involved when exposed to very high light. And then I got all bioinformatics to do a bunch of analysis after what we call the red blob. And there's 2 things I want to highlight from this. We could not detect C. apininum, that organism that was so interesting, until after we cultured. And 26 million long reads that we got from that sample pre-culturing wasn't there. But then when we inoculated, it was there. Now, to microbiologists, that might not be surprising. That is very important. And James is going to talk a little about the importance of culturing, why what we do pairing culturing with sequencing and banking everything gives so much power to the science we can do. The other thing is, I used a simple bioinformatic method to compare the sequencing from that baseline sample to every sample in NCBI as of 2022. So about 700,000 genomes, just quickly compared them all. And you can see what this is showing is the most similar thing that was ever been sequenced on NCBI at the time was that big circle in the middle, that deep hydrothermal vent. That is a very different kind of ecosystem than what we are looking at in Volcano, which is a shallow, light-exposed seep. And the Jacquard distance, the similarity, is only about like 0.17, so they're very dissimilar. So in other words, these environments are so undersampled, there is literally nothing like it that we have seen before, which is really interesting and very exciting to explore. A couple notes on our other initiatives that we have going. So, as I said, we have a coral initiative. We now have collaborators all over the world where we are trying to build this living database of the coral holobionts. So you might be familiar with this word, the holobiont. Corals are wildly complicated creatures. They are a coral, they have algal symbiont, they have bacteria, they have viruses. And the combination of those things form the holobiont, and the composition of that combination, say what bacteria are there, can confer tolerance to disease and bleaching, and all these other different maladies that corals face. Gabby will talk a little bit about corals in the context of disease in her work later on. And we're really interested in bringing cutting-edge sequencing and multi-omic methods to understanding that whole biome, because most of the sequencing that's done on corals is 16S, which we're, MetaSUB, we're not as excited about doing 16S in the urban environment. I want to bring functional metagenomics to corals, and also being able to complete all these draft genomes that we don't have, identify what are the bacteria that are living there, and then from that, use our culturing methods to be able to actually grow them out and see how they confer tolerance to different coral disease states. These are some examples of 8 different species we have now sequenced. We have another data set, a longitudinal data set of corals exposed to a bleaching event before, after, during, coming from the Red Sea. We have some samples coming in potentially from Miami, French Polynesia. So if you want to collaborate on this, let me know. Really excited to be doing a lot of coral work in the next year and building specifically the first phase of some bioinformatic pipelines for really teasing out the different genomes of the whole biomes of bacteria, viruses, coral, and algal symbiomes, all of which, as I think happened yesterday for bacteria, the reference databases for corals, they could use some updating. So that's what we're trying to do first. Now, our other initiative that we have that we're kicking off this fall is our CROPS initiative. So this is a grant where basically we're going to be going to the Cima volcanic field in the Mojave Desert, where some of the original lunar simulant regolith came from. There are plants growing there. We wrote a very simple grant. This is one of those grants to write the last minute. I think this won't be accepted, and we got really lucky with it because we had an amazing team putting it together. And the premise is we're going to go identify endophytes living in those plants, use our culturing strategies, use our sequencing strategies, and identify if they can actually help plants grow in real, well, initially lunar simulant regolith, and eventually real regolith for the next phase. And so that's going to be what we're doing. We also have a bunch of one-off projects. We'll hear a little bit about this from James, where we have the measuring the impact of deep sequencing on extremely rare environmental samples. So corals, we have interesting watermelon snow, Amox reactors. Talking about the Amox story with the people at dinner last night, it's one of my favorite stories in microbiology, predicting that physiology from thermodynamics before we had ever observed it. And so that's something that's sort of a precursor to a lot of what we think about, so it's wonderful to have those samples. We have interesting isolated bacteria. James is going to talk about some of the soil crust work that we've been doing. And again, we're taking stuff from all these different interesting ecosystems. So it's quite fun to go around the world and do this sampling and build these deployable labs. And then, okay, so last thing is where we're going. This is going to be the shortest part of the presentation because I want to make sure I leave space for everybody else to talk. As you've now seen this slide a couple of times, it's one of our favorites. This is our pipeline. And from an academic standpoint, what we are building here is something that should really resonate with everybody in this room. We call it the Two Frontiers Living Database. So what's special about this is every single sample that we take, we bank in glycerol and DMSO and DNA shield. So what that basically means, if you're not familiar, glycerol and DMSO we can use to culture different kinds of organisms. We also, in DNA shield, we use that for shotgun sequencing. Then, when we do our on-site enrichments, so let's say we throw these microbes in really high CO2 to simplify the community and select for a CO2 consuming community, we also bank those enrichments in glycerol, DMSO, and DNA shield. Then every single isolate we get out of that with our collaborators around the world, same thing. And so we then sequence all of that. And what that means is we have this massive resource that we're slowly building, and next year we'll be making publicly available, we hope, that has all the sequencing data paired with samples. So that means I'm a mathematician, right? I love folding proteins and things like that. If I find something with an interesting new variant of Rubisco that's in this weird bag that we weren't able to culture the first time, but fortunately, we put all this time into being able to do targeted culturomics based on the geochemistry and the thermodynamics of the genome, we can go pull that sample and grow it. And so that is something that we're building essentially for the academic community. Two Frontiers, like MetaSUB, is a nonprofit. Our goal is not to go start a bunch of companies or anything like that. It's really to build this and get the methods right so we can actually bring microbial biotechnology to the point where it can scale, which it's debatable if we're actually there yet. So that's something I'm really excited about. Like I said, previously we're up to a couple thousand isolates now across all the initiatives. Other aspect of where we're going, we have our fourth carbon expedition. But it's sort of carbon and coral. I've been calling it carborylon because we're sampling both of those things because I love a good word combination. And so we're going to Shikanojima. You can see these corals, beautiful place there. Also these really interesting terrestrial sites with really remarkable geochemistry and colors that, well, colors tell you a lot about the microbes that are living there. We're very excited about some of these springs, these onsets there that we're going to be able to sample with our collaborators. We're working with Sylvain Baxi, who I think a lot of you maybe know. He's local here, really wonderful, really talented scientist and great PI. And then I also want to emphasize, it's not the right button. I'll emphasize this is in collaboration with Seed Health. They're also funding this exhibition. We've got a big collaboration with Zymo to analyze a lot of these samples. I know Mike Weinstein is here. I recommend you talk to him after this. And so it's a really big team effort that spans like with MetaSUB industry and academia and all these different institutions. And so very, very excited to kick this off and continue to build more collaboration. So and last thing in terms of where we're going, the next talks that you are going to see You're going to see a talk by John Kowitz. John Kowitz, amazing photographer, freediver, very talented individual, who has basically made a really interesting career now following and working with scientists around the world. So he has documented scientists in their element and all the different environments, and I thought it was really important that you get a sort of lens, to see what the lens into his perspective of what it's like working with scientists. You're going to hear from James, who I know a lot of you have talked to, talking about more details and methods on our culturomics methods. And then you're going to hear from Davide and Gabby about these high CO2 marine ecosystems like the ones we're going to be sampling in Chicanjima, and from Gabby on some of the biophysical modeling she's been doing to analyze coral pathogens and coral dynamics. And so I really want to emphasize these talks that this is not all work that's been done by 2FP. A lot of this is people's personal work. They're going to annotate where that's the case. I wanted you to meet all of these scientists and individuals to understand really what incredible people they are and what the skills that they bring to the team. And so I can't emphasize enough that this is a very diverse window, and we took these not at all taking credit for everything, all the amazing work these people have done. But we're very excited to continue working with them in the future, and all of you. And so I want to bring this slide up one more time. Incredible group of people. I wish we could have had all of their pictures up on this. It's really, really been amazing building this, and the sort of degree of growth in the past couple years has been really fun, and I can't wait to grow more and work with all of you. So with that, I will take any questions or comments.
For your carbon capture microbes, do you know what the fate is of the carbon and what the fate becomes when they start sedimenting in large amounts to the bottom of whatever body of water they're in?
That's a great question. So we think with these ones, it's funny. I talked to like a proper marine microbial ecologist about this, this sinking behaviors, these were isolated from the water column, not the sediment. So, realistically, we do not have giant puddles, to my knowledge, of like cyanobacteria living at the bottom of the ocean. So where they're not being produced in the same conditions that we use in the lab to produce them in mass, which is why I'm mostly excited about this strain from a bioreactor standpoint, which is able to concentrate, once they can engineer it, they can concentrate biomass and do something useful with that biomass. I mean, like anything, the carbon that's being sequestered, you have your biomass production, and then of course it's cycled naturally like everything else. So it's not, if it were this like infinite sink in these vents, that would be not really how thermodynamics works. So they're part of a natural ecosystem, but by going to these ecosystems, we can identify evolved machinery that is really useful in an engineered bioproduction standpoint.
Right. Well, interesting, energetic talk as always.
Thank you.
Great to captivate the audience. That's nice. I have two questions. One is on the living database, the other one is on coral. I'll start with the coral.
Sure.
So I have seen that you have anywhere from 4 nanograms to 10 nanograms DNA that you have got.
Mm-hmm.
Most of them, 99% of them coral.
Yep.
I'm not asking you to answer right now, but we have to collectively come up with the idea how to deplete and remove the host DNA.
That is a magnificent question, and I skipped over this story because, as you can see, that was a lot of information. We have a similar origin story to the Coral Initiative as we do to the Carbon Initiative, where it was a last-minute grant, and the purpose of the grant was that. It was how do we optimize measurements, how do we deplete the coral? And so we have now spent the past 2 years with a slew of different methods, and some work really well, everything from simple filtering to target capture solution, because it is the only, as you said, the coral is 90% coral and algae, and so that 10% you have to sequence very deeply to get any mags, or you have to deplete it somehow. So that would be fun to talk about. Yeah.
So second question is your living database.
Yeah.
So here again, collectively we have to come with a way to get the metagenomes of the viable microbes without even culturing. Culturing is needed.
Yeah.
Must, because there are so many biotechnological applications that I'm a big fan of culturomics because I started my work from culture, and now I am dealing with this metagenome that have both dead and viable.
Yeah.
As a NASA person, that we come up with appropriate strategies to clean. That means you are killing, but you leave behind the DNA.
Mhm.
So somehow we have to remove that, and one of the things that we achieved is BMA. That's a big solution and everything. But to get your living database keep going, this will help you to pinpoint culture. I think Hendrickson is going to talk more about it, and then it will help you to pinpoint what the culture. So you don't have to answer, but I'm.
No, you're totally right. You're totally right with the viability issues. I mean, that was why, that was one of the first questions I asked yesterday about the preservation when you send things back, because we've had the same thoughts. Like, how do you know it's alive? How do you build it? So PMA, I think, is a good solution. That's one of those things where once we have more resources and more people and more hands on it, it'd be really interesting to sort of take one out of every X samples and throw it in there and see what we get. But totally fair point, because I mean, yeah, James led a few slides on these sort of distributions of the microbes in this data, but we don't account for what you see in the genome, what's viable. Yeah, good point. Thank you. Any other questions?
Yeah, great job, really. Well, just a couple of quick things. With regards to engineered consortia, which I'm particularly fond of, what would you say about the propensity for engineered consortia to be more of the holy grail from the perspective of you have to dump your electrons somewhere. So when you're growing on your own, you don't really have these syntropic interactions that would facilitate growth.
So this is a question where I'm glad there's not a soapbox on the stage, because I would stand right up on it and then you wouldn't be able to get me off. I totally agree that essentially, pure culture, we can talk about the history of pure culture, it is not how microbes exist in nature, right? And so I do not have data to back this claim up. I want to be really, this is my opinion based on reading papers and talking to all the people in this room. If we are going to scale bioproduction, I think, yes, the holy grail is how do we have a consortia that is working together. And ideally we can engineer, but let's just start with figuring out that it's like we talked about last night, those medium-term questions, instead of like the big holy grail of let's engineer a consortia to capture carbon. It's like, okay, how do we optimize stability and feedstock? Right? How does community complexity change over time? What are the dynamics to regress towards the mean of a single function instead of a single genome, right? Because I agree that, think about it from a thermodynamic standpoint. It does not make sense to have E. coli growing at massive scale trying to do one thing. Doesn't make sense. So I think it is the holy grail, and I do think the data we're generating will point in that direction.
Great. And so I was thinking, based on your fondness for continuous evolution, have you done anything like we've done previously, just random isolation of samples, and then just continuous evolution of communities with optimization of function as the goal?
If I had enough chemostats, yes, that would be the first to yes. So we've talked about this a lot. The answer is no, we haven't done that yet, but it is on the list, high on the list. Yeah. And I guess James is on a little bit of work, but we haven't. Yeah. So we haven't done the full. How do you optimize a function instead of genome experiment the way I want to do it.
Yes, I have one question. So it's really fascinating and then feasible for the application part.
Yeah.
So since you are actually growing the microbes all of the world, and then how do you deal with the Nagoya protocol?
Spectacular question. So we are we're Nagoya compliant and everything we do, we're very careful with it. And I mean honestly, so whenever we go into a new place, the first thing we do is ensure that we have a local collaborator that we're working with and we have representation from the actual community of people. Because I mean our goal is not to go in and take a bunch of microbes and then go and start companies with them. Like we're very earnest in our intention that we are a microbe institute, right? So what we're doing falls under academic use. We are not. So essentially, we've had a lot of long conversations, and we're doing everything we can to go compliant. We're really excited about the upcoming changes, hopefully, that make it even a little bit stricter for some of the stuff. Because there's this whole, I mean, even with the living database, I've gotten into probably ruined a few investor relationships because I've spoken a little too strongly about this. I don't think we should be clustering that genomic data off behind paywalls, but most people who I talk to say, well, why don't you just take all these samples and then your data is valuable and then you have money and then you can sell. And that's just like insulting and horrible on about 10 different dimensions. And so, from the day we started 2FP, we had conversations like, how do we make sure this is compliant with Nagoya, just ethical, and we're sharing everything we can for as free as we possibly can with everybody in the same standards and that stuff.
Yes, I think that your approach is good, but It's a international rule, so
Yes, yeah.
you need to be very careful.
Yeah, and we are, especially as we grow, it's, it's very much on our mind.
Thank you, everyone. I think in the interest of time, we're gonna have to continue. If you have any questions, just please reach out to them afterwards. Thank you, Dr. Tierney.
Thank you.
All right, up next we have John Kowitz. John is a professional photographer that specializes in capturing the underwater world. Throughout his career, he has worked with scientific and conservative expeditions to provide documentation of scientific explorations in the field. Additionally, he's an instructor in free diving and advanced scuba diver.
First of all, thank you very much for having me. I'm honored to be in front of a bunch of very incredible, intelligent scientists, and excited to talk today. I only have 3 fears in life, and then one is worms, tight caves, and public speaking, so I'm facing one of those ones too. So first of all, I've had the honor to work with Braden and several very intelligent scientists on many of these trips. So I want to talk a little bit about that. One second, this is not too fast. Okay, so I'm going to talk a little bit about my background, why you want a photographer working with you, and how we can work together to support your goals. A little about me. Growing up next to Lake Michigan, I developed an early fascination with water. Every summer we'd spend swimming in the waves, road trips to somewhere ending in the ocean. I was that kid that never wanted to leave the water. We, in high school, while other people were playing football or talking to girls on the beach, I was that kid that was at the bottom of the pool with a stopwatch seeing how long I could hold my breath. Since then, not a lot has changed. I'm still that kid in the pool. All throughout high school, I wanted to be a marine biologist. But in college, I switched into medicine and worked in medicine for a while after college. Working in emergency medicine, I quickly realized how much I like working with animals versus people. However, healthcare gave me the flexibility to travel, and I moved out to Hawaii. When I moved out to Maui, I picked up a camera to start filming the beautiful landscapes. I found landscape photography to be very interesting and fun, but I also found myself so focused on getting the shot that it took away from some of the hikes and some of the beauty of nature. However, when I combined my love of freediving, finding out underwater housing, and started diving, that was the game changer for me. I would forget when I was photographing the whales, sharks, and dolphins that I was holding my breath, and I could just stay underwater forever just filming these beautiful animals. Here is a picture of one of the first times I did a scuba dive in Kona. I think a few people here might have done this. It's a beautiful dive. I love this picture, the way the lights hit all around the mantas. You can see the gill plate of the mantas. Here is another shot. I really like the isolation of this particular manta and the way the light hits the gill plates. Mantas are very interesting. They are feeding on plankton at night, and they will come right up in your face, so they're a perfect subject to shoot. They also have one of the highest brain to body ratios of any fish, and they're incredibly intelligent. Yeah. This was my first time in Tahiti and swimming with humpback whales. It's very interesting with humpback whales. The mother is very protective, but if she perceives you as not a threat, she will allow the calf to come investigate you, and the calf are very curious. This particular calf, you can kind of see the eye contact, but it was even closer than this. And it came up to our group and just swam with us with an arm's length away. Beautiful experience, and having something like this, my first time in Tahiti was just absolutely incredible. This is actually called a fever, I found out, a fever of spotted eagle rays. They hunt the sand looking for small crustaceans and fish, and here they aligned perfectly for this photo, so I really love the symmetry of this photo. And one of my favorite animals to swim with, the tiger shark. Tiger sharks are very different than other animals or other sharks. Other sharks tend to be a little more cautious. These are powerful, they're strong, they'll get right up in your face, but yet once they realize they've lost that element of surprise, which is the main reason sharks, the main hunting strategy of sharks, they kind of just swim around and investigate what you're doing, what potential chance to, food chance there is. This is a little behind the scenes of me filming that same calf in Tahiti. So a while later, I was asked by a group of scientists to join for a big project in Cocos Islands. Hammerhead sharks are very timid, and bubbles from scuba divers can actually scare them away. So they wanted someone who could freedive spearfish as well as photograph these hammerhead sharks. So I went along with this mission to start tagging hammerhead sharks. Unfortunately, when we arrived, we found out that there was no freediving, spearfishing, or snorkeling or swimming in the park due to a fatal tiger shark attack the year prior. So we had to do everything on scuba, but we did have a very successful mission. We planted these transponders you can see here, and they're acoustic transponders that we planted all along the seamounts. The purpose of this is to relay a signal to tagged animals to essentially show the area that they were tracking between Cocos and Galapagos Islands. We also photographed a variety of sharks, manta rays, and turtles to aid in ID and show their migration patterns. So this is the Cocos Galapagos pathway, and we were working in Cocos Islands, you can see here. This is a marine protected area. The Galapagos is also a marine protected area. The problem is commercial fishing vessels will sit in this pathway. So as soon as these sharks, dolphins, whales are leaving, they get caught up in long lines, they get caught up in dragging nets, and they are many times killed just for their fins or as bycatch due to the commercial fishing industry. So our purpose and our ideal goal with this is to take these tagged animals and show where they're being caught or killed along this pathway to protect the entire pathway, not just the islands. Also in coastal Costa Rica, the wetlands are being used now to have shrimp farms, and they're destroying a lot of their breeding grounds over there. So our goal is really to protect that entire area where they're pupping as well, to make it a marine protected area. They say about 30% of all the fish we eat now is caught in marine protected areas. And when we worked with the local DNLR and Cocos Islands, they showed us their enforcement. There was 2 rangers and a tiny dinghy. And all of these shrimp boats would just sit, or commercial fishing vessels would sit right outside their boundary, sometimes coming in the boundary actually. And all they would do is try and dinghy over to them and essentially yell at them or take down their information, but there's a severe lack of enforcement and a severe lack of resources to actually follow through with these laws and rules. This trip was the breakthrough that this fun little hobby with this camera I had could actually turn into something more, and I could potentially make a difference in the world with this photography. So this is kind of the establishing shot, just showing our sharkwater vessel when we first arrived at Cocos Islands. This is another establishing shot. This is actually the coastal section of Costa Rica before we left. This is the pole spear that we would use, and we were actually successful in tagging a manta ray, a whale shark, and a blacktip shark. So we would kind of sneak up behind the shark and using the pole spear, put it right behind the dorsal fin. This is the other method that we would use to tag sharks. It's not ideal because it causes a little bit of stress on the sharks, but everything was timed, so as soon as we catch the shark, we get it on board as fast as possible, and just make a small incision in the abdomen, place this tag, and then quickly pull the hook and release it. And we tagged this, or we tracked the shark for a significant period of time, and it was healthy and doing well. So this was very successful. And this is just a behind the scenes of me filming with a big school of big-eyed jacks. Very healthy population of these out there. This was an incredible moment for us and the scientists. This is the main marine biologist, his son, and we found while we were diving this juvenile whale shark. So he was actually able to not only swim with it, but tag this shark. And they're still tagging it today as it's doing its Galapagos Cocos transition. And this is the main reason we went there for these hammerhead sharks. It's one of the few areas where there's large schools of hammerhead sharks that do that entire transition. They are extremely timid, so it was very difficult to get close to them. The next expedition, I was asked by this incredible team of scientists at SEED and Two Frontiers Project to join to go to Cocos Island, or to Vulcan Islands in Italy on a mission to document scientists collecting capnophiles from underwater volcanoes. This was a big change for me from shooting with sharks. I wasn't familiar with filming microbes, but a very interesting expedition. So gearing up for a trip like this, it's important to look at the purpose of the photographer, the purpose of the reason you want a photographer. Is this purely documentary? Is this educational? Or is this to inspire emotions and capture a documentary style film? That really changes the setup and how I want to shoot my shots. Upon arrival, we had some inclement weather, so the very cinematic, deep sights were unobtainable. So you kind of work with what you have left. We did find some beautiful underwater areas where we had some seagrass beds and shallow fissures. And it was very interesting just watching the scientific process and how detailed every little aspect is, and to watch these scientists working through the inclement weather issues we had. It's important for the scientists and the photographer to work together to decide how much time we have for photography. I know everyone, especially scientists, are very, very focused on your work. So we have to really factor in the time for if we are staging shots or if I'm purely a fly on the wall, just trying to document everything that the scientists are doing. Also, communicating underwater adds another challenge, but for me, really trying to capture that action and show the beauty and scientific nature of what is going on. Again, it's very important communication for the scientists that we may not understand certain special situations, such as a sterile field, potential for sample contamination when we need to stay back, or as James informed me, do not pee in your wetsuit, it close to pH monitoring site that can throw off our samples. This is the amazing team in a prep day, getting ready for collecting all the data ahead of time. This is this incredible scientist preparing all the media in a very sterile environment. This is really nice to be able to establish these documenting shots of the area that we'll be working ahead of time. And this is this active volcano that was still spewing these sulfur clouds. Beautiful backdrop for all the science here. An outdoor lab that was set up with that beautiful backdrop, collecting samples as soon as they would come out of, or processing samples as soon as they would come out of the water. So our team of divers getting ready to descend. And it's shots like this really paint a picture of everything that we're doing. This is Braden, and this is what we were working with, about 6 foot of water, so a little different than what I was expecting originally. But these are the CO2 bubbles coming out of the underground fissures here. I thought this was kind of a beautiful shot, just showing the bubbles and the scientists working together and the very detailed documentation that they were going through with every set of samples here. Working with what you have, I loved the look of this beautiful seagrass bed with the light coming behind the diver as they were moving towards the next site. We had a little challenge here with our sample tubes being full of air. Obviously it floated, so we put them in these bags, but it actually added this beautiful color. This wasn't planned, but I thought it was a really cool shot with the bubbles and the colors, and then finding a new site to sample here. So as excited as I get when I see a beautiful shark or a school of dolphins, it's pretty incredible to watch a scientist get excited about finding a new bacteria or microbacteria. So I loved James's excitement when he is looking through the microscope finding a new bacteria here. Fast forward about another year later, I was able to join them on a trip to the hot springs. And this was a very different trip, no underwater here, but a very beautiful trip. And that's kind of an establishment shot. Here, we kind of showed the journey. Again, I like to really tell the story of the whole expedition. So this is showing the journey of the process to get up to these hot springs. I really like to include some of the trials and tribulations that we went through along the way. This particular day when we got to this hot springs, it was freezing cold and we got caught in a blizzard. It was very challenging to get samples, but the scientists pushed through. I was probably the first that wanted to go home and not push through the situation, but incredible to watch them all work in these harsh conditions. Besides the painting the picture, it's very interesting to get up close. Here, looking at the texture of this algae in some of the hot springs, I thought it was a really cool shot just to get that macro perspective. Again, this is Braden collecting samples at Pinkerton Hot Springs. The sulfur turned the area, or this algae, into this beautiful orange color. And you can see on the next slide here, that close-up macro shot of this, I thought was just incredible texture, color, and I really like the look of this. A big change in me from the usual photography I'm doing. So why do we want to document all this through photography? Photography is a communication tool to bridge the gap between science and aesthetics. So you got people talking, so you got people thinking, and ultimately, hopefully, to get them caring. The majority of people don't have access or don't see what a lot of you see. They don't see the damaged reefs, the rising CO2, the destroyed rainforests. So adding photography or videography is the visual storytelling to bring attention to our cause. Media can paint a picture of the research, and good photos can add to the campaign, to the publication, or to the documentary. Also, being able to look back at a comprehensive collection of photos in the future allows you to really paint that full picture of the expedition. I recently watched the biographical film about the explorer Sir Ranulph Fiennes and his life and exploits. If anyone hasn't seen that, it's a great documentary film. But seeing all of his pictures of the explorations in Arctic and the old videos really gave life to the entire story of his exploits and allowed this film to be made. And finally, the picture is worth 1,000 words. Has anyone here seen this picture from about 2015? So since 2015, this viral video of this straw extraction has garnered about 110 million views. Since then, 30 countries have banned single-use plastics. Companies like Starbucks, Disney, Alaska Airlines, and McDonald's have phased out plastic straws. The turtle has become a symbol of plastic crisis. But the straw only makes up about 0.03% of plastic pollution. A large percentage of that comes from the commercial fishing industry. Here you see an all too common sight, ghost nets, which either can be discarded from commercial fishing vessels or broken off from their anchors, float throughout the ocean, collecting things like this, turtles, sharks, dolphins, and it's an all too common sight when we're out diving. About 60 to 80% of what is caught in shrimp trawling nets is bycatch, meaning these animals are unnecessarily killed many times before they can be released. They pass away in the nets. So showing a picture like this versus showing statistics can have a huge impact on public perception and what we want to do to make these changes. Here you see our brilliant scientists in action. Again, talking about planning for these expeditions. So really knowing local laws, rules, and regulations, knowing the weather conditions, thinking like a photographer, and the fact that when to use your lighting best, how much time you're gonna have at sites, how much time we need to science versus how much time we need to pose a shot or to redo a shot for the scientific documentation purposes. Again, do we want the photographer involved up close, or do we need them back away to not ruin any science that's happening? Action shots, we want to really overemphasize something we're doing. So something that may be trivial, really extending down to turning the angles or making it look very cinematic, and we can work together ahead of time to make that look good. Showing the epic landscape where you're working. A lot of times we maybe take for granted how beautiful the landscape is. So having that in the background, really showing where the science is taking place, can paint that beautiful picture. And we also want to capture candid moments. So use shots that best support the goals. Take the time to get the shot. This is also true in weddings and in engagements. It's usually the groom. They want to be done with the shots, they want to focus on what they're doing. But in the future, taking that little bit of extra time to show what you're doing or that beautiful landscape can really, you'll thank yourself in the future for taking that little bit of extra time. Again, finally, people sometimes get nervous what they're going to look like in camera if everything's perfect. But the nice thing with digital cameras nowadays versus film back in the day, if you don't look good on camera, or if something isn't quite scientifically accurate, we don't use that. So we can always pick and choose at the end what looks best to support our goals and missions. With social media and digital presence ever on the rise, it's become increasingly important to have a presence, to bring awareness to your research, and to show what you're doing to the public, to potentially bring attention, to collaborate with other scientists, or for funding or documentation purposes. SEED and True Frontiers have an incredible group of scientists really supporting my photography, but also just working with, for the first time, working in the scientific field and really seeing how they operate. It's been incredible to work alongside them, and I appreciate all these opportunities. Thank you.
Thank you very much, John. Any questions?
Okay, I have a question. So, really beautiful pictures. So I'm personally very passionate about scientific communication and outreach. And something that has been on the conversations within the scientific community is, are we as scientists doing enough to communicate to the general or normal people like that are not like scientists? Do you think working now, like from your past experience working with animals, like in your own work, and now being in a closer look with scientists, do you think scientists are doing enough to bring that work of what we are doing or what we're trying to do to just the general population, or If so, or if not, what do you think we should be doing better?
That's a great question, and I do think there's a big gap there. I know scientists don't spend days scrolling social media, and you're focused on everything you're doing. Rightly so, but I think with, like I said, the digital presence, there's a lot of potential collaboration that can happen. And I found this, how much I want to get into this, but there's an amazing group of scientists that work in Costa Rica and in Hawaii, and they're doing amazing work. But there's one influencer, I won't go into names, but who's a big shark diver, and she has grown a huge following, and she's got tons of funding and can travel all over the world and do what she wants. But she's not really doing the research and the science, but she has the funding because of her social media background to have the resources to kind of do whatever she wants. So I would like to see more actual scientists who are really doing research and making a difference be put on the forefront versus influencers, and really have the resources given to those people who are making a difference in the true science to have those resources.
I have a question for you, John. So I think that sometimes scientists don't realize the value of showcasing to the world, the importance of the research. And I'm curious, like you from an artistic perspective, what has been some of the challenges of like teaching the scientists, like, hey, this is something that like you should be caring about too? I don't know if you can speak a little bit about it.
Yeah, I think I think James is a prime example of this. But I love it. He's so focused on the science and so focused on everything he's doing, and pictures are this nuisance that kind of get in the way. But I think once you look back at some of our expeditions and you look back at the photos that Brayton was using, you kind of get to see it. Okay, this actually was important. I'm really glad we took the time to do this. So again, like with weddings and engagements, it's annoying, but when you look back years later and look at that beautiful time together, that wonderful memory, or that piece of science that you collected, I think it shows the value of how important it is to take that little extra time to capture those amazing moments.
Thank you very much for this extremely interesting talk. Very beautiful pictures. I do see what you're talking about, that is the gap between what science is doing and the general public, and I like the link that you made. Do you think that this is a teachable skill? Because the same way that a tool cannot be used if it's not user-friendly, science is not communicable if it's not clear, if people cannot understand what's being done and the relevance of that. So you think at least to my knowledge, I don't know if there is a certain course that you can take or a way that you can learn this skill of communicating science. Do you see yourself someday teaching your perspective how to communicate science? Do you have some insights into that?
Well, now you gave me a good idea. No, I think there is a huge gap, I think, between art and science, and so putting those two together has a lot of potential, and I think they can help each other out so much. So that is a big gap, and I think that should be something that should be taught to really aid in what you're doing. Sometimes this amazing work can go on the wayside or not be brought out to the public. So putting those two things together is a big thing that needs to happen.
John, to add to what Maria was asking, every single agency that is funding has 10% of the money kept aside for outreach. So each PI who got the grant can tap that money, work with the institution, don't come to you, works with the institution to promote their science. So to answer the next generation scientists like Maria and others and Brandon, they can go back to their agency. NASA is doing it for sure. I don't know for NIH or something, NASA is doing this. They keep 10% of the money, separate the budget. If you are getting $1 million, 100,000 is set aside for it. So
to echo that, I know the Department of Energy funding is just really good, very sound science communication team that can help do exactly that. And I do want to say that the scientists, I think we're starting to realize the importance of communicating what we're doing to the population. Yes, when I'm out in the field, I think it's very focused, but we're also, scientists, we're always supposed to be learning and learning from our mistakes. What is science, but learning from our mistakes. And so actually, I have learned a lot from John about how people respond visually. We're very visual creatures. And so this kind of artistic capturing of the science to communicate, I think is super important. And I've sort of come around to carving out the time and effort to make sure that we're capturing these images to give back and help the funding agencies and the funders, the companies that are funding our work, and the foundations, like they need to reach out to the public as well. And so that's something that you can be providing for them while you're doing the science that they have given us funding to do.
Yeah, scientists do the scientist job and photographers do the photographer job. Otherwise, you cannot excel in science.
Yeah, nobody wants to see my photographs.
All right, thank you so Sam. We're gonna have to cut the questions short, but I invite you to come talk to the speaker during the break. We'll reconvene at 10:30. Big round of applause to both our speakers today. Thank you.
That's what I want. I want the deep cut one person. It's not fun. Yes. Yeah, well, that's what we talked about. I mean, I talked about it.
But then I came back, I couldn't find I work with country.
So that's why like we are
Yeah.
well, actually, you can modify. You can modify a single base.
Yeah.
That's something.
But not fully.
Yeah, but it's like how do we engineer it? Let's build a bunch of. That's what. Or like using this thing.
But I want to know.
And they also like fingers, they actually.
What I want to do is
I think so, yeah.
find these microbes and see like the surveillance, like the water microbes.
wasn't inclined to like actually use like, I'm working on like apparatus or trained or different ways to simulate the gut to then allow me to function. So I'm like excited for Jim to talk. And it's like, yeah, I feel like
For real, like, it's people with the carbon
yeah, it's great. So you have
Such that it's stable. Like, think about a normal distribution of stable distribution is the reason why. Set three. And so I think we should take that little more time to grow.
It's interesting with older companies that it's bringing, the ecosystem. Right?
You have to also get like a copy of this. For like finding a way to capture safety. Yeah, but do you wanna like transform?
Oh, you mean? Yeah, so like, this, like, this. That is like, once we it's exciting.
We have 15-minute break. We can meet at 10:30. Regular lunch break.
So like,
Yeah, one of the
my question is, what is so good or like interesting about the process?
So there's two things.
Okay. I said, do you have access to this other file? Okay, so you only have access to these questions. Okay, so then there are some, so these other files, how and
So how
and
we
then
can,
changes.
I would then.
Yeah, so we can then go through this together.
Would you
Okay.
hear that or like, because syncing, everything is just too big.
Which is not like a you produce that, but you then basically sort on it. And so it's tremendous that it's like not very awesome subject. So whatever. So it's a different application space. That's
Yeah. One thing I do know is that like biopreserved must be made in microgravity conditions.
There's a dynamic component to like about sedimentation, right? Like when it like doesn't go back up after.
Oh,
yeah. I thought I read that somewhere from one of the stuff.
I mean, it's possible next to that experiment.
Yeah, actually, same. I'm wondering how you can like use the syncing nature to like the biofilm.
Yeah, I just think it's a cool
Making
trick.
of it
I just want
to an
it's a
advantage.
fascinating. I don't know. Like, yeah, we do it. We do it.
You got it? What? Oh yeah, he's gonna download it. Or he's gonna. Hello, live people.
Yeah. Who's there? How many?
Hey Scott.
Yesterday they were saying this is a little bit, they couldn't see the slides. Should we move it that way? A little bit.
You could, yeah.
Actually, it's live, so.
Okay, hey people, we're gonna move you.
It's gonna.
Maybe here. Yeah, I think it looks better. Yeah. Yep. Yeah, looks good.
We have to twist it. No, you can't get it like this. You just put it like this and you twist it.
I don't like it. Electronics. Okay.
Carlson's in the afternoon session, right? Right before.
Yeah.
I just forwarded the Dropbox link.
Oh yeah, you should. Well, yeah. I know I'm getting. I have one of the yeah. That's your point.
This one is our second edition. This we're gonna have metal, very thin metal one. Edge on, yeah. Exactly. That's what I was talking about.
Oh yeah, yeah, yeah. Is the experience?
All right, everyone, we're gonna get started in the next couple minutes. You're gonna take a seat.
Yes, 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 yes.
All right, everyone, we're gonna get started. The next session is a talk by Dr. James Henriksen. Dr. Henriksen is a microbiologist and bioinformatician holding an appointment at Colorado State University as a research scientist. His current research focuses on the exploration of microbial resources as a co-founder and director of R&D at the Two Frontiers Project.
I want to thank everybody for all of the great science that you've been sharing with me, and I feel very honored to be invited to this MetaSUB conference. Thank you. I want to start a conversation about what I'm calling next generation culturing, by which I mean using some new piece of technology, using some ways of accelerating traditional environmental microbiology, and combining that with metagenomics. Because I think, and this has actually come up a number of times in this conference and in conversations, I think there's a lot of benefit of trying to find these crossovers between these two areas of expertise, and I think together they're really a lot stronger than they are independently. So we all know that we live on a planet dominated by microbes, this vast diversity of the microscopic world, and all of their amazing capabilities that are key in the carbon cycle, in breaking down and forming an immense number of different compounds and transforming elements, respiring iron, capturing light energy. And I really love discovering those functions, what organisms do. And so I want to just talk a little bit in the abstract about the strength of culturing when you care about the functions of the microbiome, and then go into a couple of stories that illustrate, I think, different ways that these two areas of expertise can collaborate. And the first is finding new genes and new functions of known genes, because so much of bioinformatics depends on what we already know as captured in various databases. I want to tell you 3 quick stories about how you can use culturing to find emergent functions of microbes or microbial consortia that can be very difficult to tease out from purely bioinformatic methods. And then I want to kind of flip it around and tell you some ideas in some interesting directions of using metagenomics to improve how we culture and grow organisms. So this diversity of microbial function had some, like anammox, have been discovered mostly because of the capabilities of next-generation sequencing, but a lot of what we know about the microbial world was from environmental microbiology, where you go to a place that has a particular function, you sample it, you enrich it. And when I say enriching, I mean something very specific. I mean putting an environmental population of microbes under specific conditions, often culture media, or particular kinds of light and temperature, and having a subpopulation that together has a function of interest increase in abundance. It's still a mixed culture, it's a mixed community, and even some of the things that from your original sample are still there, just not being amplified. And in traditional microbiology, you would then go on to isolate and get a clonal pure strain. And I think that there's some interesting applications of metagenomics where you don't necessarily have to go all the way to an isolate to get some really interesting information from these kind of mixed culture enrichments. And the history of environmental microbiology, one lens, is to see it as a series of improvements in ways of thinking and different approaches of how to grow organisms. And I'm going to touch on a couple of these methods about matching the habitat and about using higher throughput. What culturomics means to me is just you try, hopefully, an informed large number of different approaches to capture a larger swath of the microbes that are in a particular environment. And while metagenomics and bioinformatics is the foundation of how we approach things with Two Frontiers project, we've also been really intentional in designing in these enrichments and these isolations to address particular functions or particular scientific questions or application areas. And in this audience, I don't need to talk about how awesome metagenomic sequencing is and why we sequence things and how you can get great information from that kind of data. But I know some people in the audience know very well why we culture and are very familiar with it, but I think some people maybe have been thinking more from a bioinformatic and genomic method. So I want to emphasize some reasons why you might want to culture. One is you can amplify based on function, like those enrichments that I was talking about, and you can be incredibly sensitive per billion levels. It's very easy to get a single isolate from a background of other organisms, which is very difficult to do when you're trying to tease out a mag of a very rare organism. You can make experimental inferences based on inexpensive assays. Sometimes just the fact that organisms are growing and making the media cloudy can tell you something about what's going on. And you can discover these emergent functions, as well as banking material for future applications and work. And so sometimes it's a little bit hard to think about when you're thinking about sort of culturomic approaches, trying a bunch of different things. So I did a little Monte Carlo simulation. In nature, many natural systems, microbes are distributed log-normally. And what that means is that this very long tail of very rare organisms, and this is part of why metagenomic sequencing is hard. And I just ran a little simulation. Say you do 20 enrichments and you assume 1% cultivatability on each media. And what this does is it amplifies things randomly very far down in this long tail of the distribution. And with metagenomic sequencing, you might get good mag assemblies for a very small number. And long read does help with this. However, just simply growing a subset of the organisms that you care the most about allows you with relatively modest sequencing to reach very far down into this long tail. And Braden mentioned how even with pretty deep sequencing, we weren't able to detect the cyanobacteria in samples. We now know it's there from further work with some chemostats. So let's go into the actual, that's all theory. Let's go into some actual examples. And this first one is from my work at Colorado State University. The rest are all Two Frontiers. Before I can tell the story, though, I have to do a little refresher on some organic chemistry. Because of the chiral nature, the handedness of the carbon bond in sugars, there are many different conformations. They're exactly the same chemical structure, same amount of energy. However, they are molecules that can't overlap, just like your right and left hand can't overlap. And to chemists, DNL sugars are indistinguishable. But to life, all life has a handedness, and since the time of Pasteur, it's been dogma that life only interacts with D sugars. It's biology, there's an exception, the vitamin C synthesis pathway, 1L sugar. But otherwise, everyone just said that only D sugars interact with life. This has been used as a biomarker for life on other planets. And we're all familiar with glucose, and it's true. The largest number of genes and the largest amount of sugars in nature are D sugars. There's a few genes that are known for a few of these other more common sugars. There's that exception I told you about. But the vast majority of them, life is not known to interact with it. There's no genes. And humans have taken advantage of this nature of sugars. There's an alternative chiral sugar sold under the name allulose, or L-cyclose, same name. And it's used as a low-calorie additive because it's the same carbon bonds as glucose, tastes sweet, but yet humans can't use it, so it has no calories. And the foremost research institute for L sugars is here in Japan, and Ken Izumori is to my knowledge, the only person who has ever tried to find organisms that interact with all sugars and actually invented biochemical methods to make that alternative chirality artificial sweetener using side reactions of some thermophilic enzymes. In this whole paper, though, they found 4 organisms that could grow on L sugars, the form of sugars that life isn't supposed to interact with. And one of them, they went all the way to discovering the genes responsible. And the key gene there that deals with the alternative chirality in all of our bioinformatic pipelines would have been annotated as involved in breaking down a cyclic carbon from plants. Nothing to do with weird sugars. So with some collaborators at CSU, they had these early hints that there was actually a lot more transformation of these rare sugars in nature. And so we set out to find the microbes that carried out those transformations. But how can you do this? You can't use all of our wonderful tools of next generation sequencing and metagenomics because we don't have any genes to look for. However, you can feed organisms just that one sugar, this alternative chirality, the L sugar, and if they grow, they can make cell material from that sole carbon and energy source. And so over a summer project with some undergraduates, they collected 150 environmental samples from all over the United States of America, all types of samples, soil, water, fruits. We tried to follow some of these hints in the literature that some plants make some rare sugars. And for every single one of those samples, they grew them on every single one of these rare sugars in this list. That's 3,600 tries. And you want to give students something they can be successful at. I wanted a large number because I didn't know if we were going to get anything. I mean, 4 known organisms. I was hoping maybe we'd find another L-glucose degrader. Every sample we tested could use these rare L sugars. Every single sample had at least one that it would grow up on of these things that life isn't supposed to interact with. And including the built environment, we didn't have very many of those samples, they're underrepresented. So, what are those microbes that are growing? Well, I couldn't grow them on agar and get isolates, and I suspected that some of these compounds might be broken down by multiple organisms in the consortia. If I grew them on petri dishes, then organisms would just grow on agar. Some of these are very slow-growing organisms and hard to get in culture. So I use an old method of where you dilute your material until there's a very small number of cells, and then in the next dilution, none. And then you let them grow. And then you take that grown-up cells and you do it again and again. And so that's a lot of dilutions. Doing this 3 times from those positives with multiple organisms from multiple samples on each sugar, we were able to recover 102 cultures. Most are pure culture isolates, some are mixtures of organisms. And we've now sequenced all of these organisms. And this was done in the same one summer by a team of undergraduates because of the capabilities of relatively inexpensive liquid handling robots. Students programmed this device and it carried out the dilutions. We had very careful controls for sterility and methods of sanitizing it. And it's really an enabling piece of technology. These things aren't just for DNA isolations. You can use them for repetitive tasks because people's time is valuable. So what are they? Is there special novel organisms that degrade L sugars? Well, actually, there's a few oddballs scattered in here, but many of these genera, at least higher taxonomic levels, are exactly what you'd expect to get from minimal media from these types of samples. And there's no clear pattern of particular taxa tied to a particular sugar. But we only had tested each of these on one sugar. I wanted to know if they were specialists in a particular rare sugar or generalists. And so for next summer, every single one of those 102 cultures was grown on every single one of these different components. So that's 7,000 odd experiments. And we were measuring their time course daily so we could see the difference between organisms that grew up very quickly versus on other sugars, the same organism would grow up slowly. And we did this just in a typical MetaSUB plate reader, because these organisms grew slowly, so we just had to measure them once a day. For faster growing organisms, there's now these inexpensive light sensing devices that communicate over Wi-Fi that you can put in shakers to measure. And you have to be really careful with things like humidity and fogging of them. But they really allow some pretty cool science to be done. And you can generate these different curves and start looking at patterns. And you can look at phylogenetic patterns that actually have hints of the mechanism. So lots of things grow on a lot of these different rare sugars. If you look up in the rhizodiales, there's sort of a bunch of sugars that are pretty common across them. And they all share, the sugars share chirality at certain points of certain carbons. So I think that's a 5' oxo reductase. And there's another one down in the Mycobacterium, it's a cluster to the side, that here, that is they all share an unusual chiral sugar at the C3 position. And there's evidence in the genomes that it's a novel C3 telomerase. And we're confirming all of this by heterologous expression in enzyme assays. So that's a story about how you can detect new functions for known genes and discover totally novel genes simply by asking an organism to grow on a compound. And I want to tell you a little bit about these higher level functions. Something we're interested in for the Crops Initiative and for other work is EPS production, exopolysaccharides from cyanobacteria, fixing CO2 out of the air and making these gelling polymers. Now, there's some genetic determinants for EPS production that you could detect bioinformatically. There's spore-like states of cyanobacteria, but it turns out that many of the soil and cyanobacteria don't have any determinants of desiccation resistance that you could detect easily. And we really wanted to get this kind of emergent function. Can an organism survive drying out because of EPS production, which is very difficult to do bioinformatically. However, you can just take your sample and let it dry out and grow it up, and then let it dry out and grow it up. And only pass those that make goo and are slimy. You can see with your eye EPS production at the levels these cultures are making them. And so you can, my arrows got messed up. So you can culture these organisms and then you can go and sequence them. And we did that. And if you look carefully, there's a lot of little dots around these cyanobacteria. We were able to get them in unialgal 1-cyano cultures. The one on the top has 2 different cyanos. But you can see this entire universe of other organisms, heterotrophs growing on the EPS and in symbiosis with the cyano, which I think of cyanobacteria as the trees of the microbial world. If you pluck a tree out of the forest, it's going to come along with all the insects and birds living in it. It's very difficult to separate them. However, we don't need the pure culture of the cyanobacteria to do this work. You can sequence them, and it's very easy in these more defined communities to bin out the cyanos and the other organisms to look at taxonomic definitions of the mags, and we were able to find novel EPS long biosynthetic gene clusters that were complete and have some really interesting novel genes in the organisms that we already know are desiccation resistant. So here's another story about emergent function. What we like to do is be able to take something in the whole biome that's producing antibiotics that we can then use to inhibit diseases. And I think finding biosynthetic gene clusters in metagenomic data is incredibly difficult. We heard some great talks yesterday about people who are successful at doing this, but what's even harder is telling me, oh, this biosynthetic gene cluster inhibits this specific coral pathogen. That's very hard to do bioinformatically, but I had a really talented undergraduate who was part of an honors thesis did this. Isolate organisms on media that we designed, tease out specific groups from the coral high-level biome, and then you just grow up a layer of coral pathogen. We're using S-marcescens as a model for that in this work, and dropping your newly isolated organisms on it. If you see a little zone of clearing, you can see in some of those images to the far right, you know that that organism is inhibiting the growth of S-marcescens. And then you can sequence them and find the pattern of activity of the antibiotics that these organisms are producing. last story about this emergent function from the carbon-1, Brayden told a little bit of the story about how we were able to, because of the collaborators, biogeochemists, during geophysics, talking about some of this, we were able to design these media so that they represent the environment that the organisms are already growing in. And then I tweaked out the media so that it would have higher growth and only allow particular physiologies, like the organisms that could capture CO2 with light or capture CO2 from the oxidation of sulfur, which we think is important in the system. And we were able to see these cultures grow up from not all, but many of these different events under these conditions. And then this is banked, sequenced, and we're isolating organisms from this. Brayden already told you about the sinking, rapidly growing cyanobacterium, and I wanted to just put in a little discussion here of some enabling technology. So in order to test all of those conditions that are in that paper, this multi-cultivator was used. And that is a device that really allows you to do these kind of manipulations. We also had some work on a sample where we passed them through a chemostat and we were able to amplify up this fast-growing cyano. There's also the ability to use these kind of bioreactors in chemostat modes where you can add in and remove material and very carefully craft conditions for specific metabolisms and growth rates. And there's this explosion of small open source and commercial microbioreactors. And the reason I'm excited about these is because large bioreactors are big, expensive, you can't take them out to the field, and I can't afford that many of them. But these small ones, I can run a bunch of parallel experiments, and I can try a whole bunch of different conditions simultaneously. And so I think they're a really enabling technology from microcontrollers and miniaturization of various sensors. The laser OD on that one. So lastly, I just want to flip it around and say the kind of metagenomics that MetaSUB has been so successful with, I think can really help culturing a great deal. And we've heard some interesting approaches of using metabolic models and interactions to start to tease these out. And I'm really excited about something we've started doing from the Colorado Two Frontiers expedition. So these features vary wildly. There's more high CO2 springs in Colorado than the rest of North America combined. And they're wildly different in their geochemistry, and they're criminally understudied. And you can see just from the coloration, the sulfurs and the irons, and that one's acid mine drainage, like these different springs are very different. And when we went into it, we didn't have that same kind of depth of geochemical knowledge about every spring. And so I couldn't just match the environment that these organisms were growing in. And so what I did was throw a lot of things against the wall and see what sticks. I tried combinations of iron, nitrogen, sulfur, light, not, such that in each experiment, there was only one source of potential energy for organisms to grow on. It was a highly selective minimal media, but I was trying 36 of them. And it's fascinating, from a single carbon spring, inoculating them into this matrix, you got wildly different enrichments and biofilms forming, even things that are unialgal in one try. And there's some iron oxidizers down here, organisms that seem to be carrying out some interesting iron-sulfur transformations. And so this is helpful to then move on to other enrichments, but we're also banking and sequencing all of these. And what we're doing with that is we're building up a database that when you grow under a particular set of conditions, these genetic sequences amplify. And what I love about this is it's completely blind to what we already know. I don't have to look only for sulfur oxidizing genes that are known. Organisms that grow under conditions where sulfur oxidation is the only possible source of energy have to have genes that are carrying out that function. And so over time, we are amassing a database that will allow us to sequence things in the field and immediately know what part of the combinatorial matrix of media to grow and enrich that part of the community on. So I think that there's a lot of value in combining sequencing and culture-based, and I was really impressed with a lot of the discussion and interaction that's been going on over this conference about people who are doing this thing, or who are experts in one side or the other coming together. I think there's a lot of strength in that. We should do more. I'm really geeked out by a lot of these miniaturization technologies that make some of this experimentation possible and affordable and less laborious. And the final thing is a very practical, specific ask of the community. It's not always possible. Low biomass can be an issue. How you cryogenically store things might depend on the kind of question you ask, but please consider every time you take a sample, cryogenically preserving the living organisms. We care about life. And so if you do that as well as collecting DNA and sequencing, it'll open the door to these kind of collaborations or future work where you can use the power of culturing and sequencing combined to achieve great things. So I want to thank the folks who really made all this possible, my collaborators at Colorado State University and the Climate Solutions Group, and our funders, two foundations that have been very generous in supporting this kind of exploratory work, and the undergraduate student teams that did all of that work discovering the L-sugar, alternative sugar genes. Really amazing group of folks. And I want to thank everyone involved in the wider Two Frontiers community. On this slide and not, I feel so much joy and privilege of being able to do great science with so many great people. I'd be happy to take any questions.
Thank you very much, Jory, for the nice talk, and thank you for educating the informaticians how important is biology, that culturomics is very, very important. Without that, we cannot build a new database, and their DNA sequence cannot be compared. It goes as uncultured. Anyway, so I have a couple of things that suggestions more than questions. That obviously, if you are growing these things, it seems that cyanobacteria is the main thing that majority of the slides I'm seeing. But what I'm trying to see that if you grow them in their own extract, for example, if you're going for soil, so take the soil, get the soil extract as its own thing, and then try to grow.
Yeah, and both those methods and the idea of actually putting chambers in an environment to grow these organisms were on that slide of different approaches, and there's certainly some questions that's a great answer for. I'm very interested in applications of these organisms, and so what you don't want to do is get yourself trapped in a place where you can only grow something with an extract from that place. And there's some tricks for this. For instance, for soil microbes, you can use brands of potting soil that put in inordinate effort to keep their potting soil very chemically and geochemically consistent. So you, in some ways, rather than using extracts from the place, you can use these chemically similar extracts that you can ensure some consistency so that you could in the future grow up, large amounts.
That's a secondary scale. You already shown, once you grow them and if you're getting it, then you can go back to your synthetic medium that is very simple that you can do it. And that way you can remove all the other dominant microbes. The other one is, obviously some of the microbes will grow together. They cannot be separated.
Yes.
And
I always try to design my experiments to allow that to happen. One of the reasons that I find Petri dish streaking to be a somewhat limited technology is because it really selects against consortia and syntrophies. And we know that that is a dominant mode of interaction in nature. And we were just before this talk, I was having a great conversation with class about some areas where that seems to be the only way that nature accomplishes certain processes. So I just allow that to happen. That one of the nice things about dilution to extinction is that organisms that interact, you end up keeping that community, even if they aren't physically attached, because once you pass them into a dilution where there's only one of the members, nothing grows.
The biggest concern I had is with the coral, the epibionts and symbionts cannot be separated, and you can even visualize this.
Yeah, so for that one, we're taking what you were mentioning, this idea of using extracts from the environment, and actually on the island in a few weeks from now, we're going to be using some media that are designed to provide some of the things that the coral and algae side of the symbioses provide to their microbiota, and see if that increases our rates of success of growing some of those organisms. Thank you.
Yeah, I'm very fascinated with the rare sugar aspect. Specifically, I was curious if when you looked at the bugs that could grow on these L sugars, did you compare them to their growth on their D and chiral?
Yeah, so this is actually, that's a really fascinating question. So to chemists, D and L sugars are the same because they carry out the exact same chemical reactions. To microbes, D-glucose and L-glucose are actually more different than D-glucose and any other L-glucose sugar, because the reason that it's L is that it's opposite at every single point of chirality. And so that's part of why I ordered those sugars in that way. You'll notice I didn't put the D and Ls next to each other because it actually obscured any relationship there. But to answer your question more directly, I did not try these organisms on every single D sugar. However, they all grow on D-glucose, and many of them grow on D-mannose and some of the other more common D sugars. And I find that sort of interesting from thinking of it from an evolutionary and ecological perspective, because I think what that means is that these rare sugars, they maybe aren't all that rare. There may be sources of them in the environment that we just don't know about yet. It's very difficult for chemists to measure these. Sometimes when they're measuring D-glucose in a plant or in the soil, it could be L-glucose. And so I think that if I found a bunch of organisms that were specialists on one rare sugar, I would think that maybe there's something special going on there. But finding it so widespread, just that no one had ever looked for it, makes me think that really there's a lot of these different chirality carbon compounds that organisms need to break down. And they are formed at low levels in things like forest fires, because that's a free radical chemical process and makes every possible carbon structure. So I think that there is sort of why we're seeing this pattern.
Yeah, and then one more question, sorry. Would you expect them to be like used simultaneously together, or do you think it'd be like a hierarchical situation?
I need to pull out some of my small chemostats and do the dioxic shift experiment on them to answer that. I don't know, and it wouldn't be fascinating if some of them preferentially used. I mean, and there were ones that grew to higher density on the L sugars than on glucose. Some of the monads. So that'd be what a great question. Thank you. I'm going to go home and do that experiment, and I will
Right. We have time for one more question before we have to continue.
James, real quick. Sorry, I have a microphone first. Great talk. I loved it. A couple of things. Most people don't realize that chirality, when it comes to microorganisms, was Louis Pasteur's initial work. So when you threw up Lunagradsky and Bayer and all those things, you should have had Pasteur on the slide for chirality.
I'm less of a fan of Pasteur as a person. And I did at least give him the name check when I was talking about chirality, because it was his initial work with tartaric acid that allowed people to discover that chirality exists, which all of us as microbiologists, we can use when we're speaking to chemists, because they get very upset that biologists figured out something about chemistry before they did.
Great, thanks.
All right, thanks so much, James. All right, up next we have Dr. Davide Spatafora. Dr. Spatafora completed his PhD in marine ecology from the University of Palermo. His research is driven by understanding the impacts of environmental changes such as ocean acidification and global warming on marine communities. His expertise spans diverse marine ecosystems, from fish to coral reefs.
Thank you for the introduction. So, start to say thank you to everyone to invite me today, and also give me the opportunity to talk, even because I'm not a microbiologist, I'm not like a bioinformatics. So I'm pure marine ecologist. So, but that I think this is the point. So bringing like scientists with different skills, share knowledge. So this is the most important things. So I'm like, as I said, marine ecologist. I'm based in Palermo, Italy. So working as a synodology Canton Dorn. And I've been working like, I got a PhD in marine ecology at the University of Palermo, then I moved to Japan at the University of Tsukuba, Hashimoto Marine Center. And during my career, I've been working on trying to assess the effect of climate change on the marine ecosystem using some unique specific environments that are called like, in different ways. So we have high carbon dioxide marine ecosystem, what we call also natural analogs. So there is many ways to call them. So try also, because they can also give important nature-based solution. So to mitigate the effect of climate change. So the climate change, I think you already know about that. So the rapid increase of anthropogenic CO2 in the atmosphere is causing the increase of the global temperature, ocean warming. At the same time, this huge amount of CO2 is solved in the seawater, is like creating declines the pH, increase the CO2, which is best known as ocean acidification. So, but this kind of phenomena like acidification, warming are altering the marine life. So, and are expected to alter the biodiversity of the marine ecosystem. So they have different like effects, and they have effect on all the level of biological organization. So from cellular, from organisms, population, up to the community and ecosystem level. So for the population organisms, so what we found there is a huge like diversity in responses. So there are taxas that are more tolerant to climate change, taxas that are more like susceptible to climate change. So there is a huge diversity. For what we don't know like very well is the effect of this climate change at the higher level of biological organization. So community and ecosystem. So, but there is some generalization about that. So there is like changes in habitat complexity, change in biodiversity, which can have effect on on the ecosystem resilience. So, but the problem is most of these studies have been conducted so far using single species, like the short-term experiment under laboratory condition, which may potentially underestimate the ability of the organisms to adapt or acclimatize in the long term to ocean acidification and warming. Also, without taking in consideration the complex interaction between the organisms and their environment, because when we put these animals inside, for example, the laboratory, we cannot take consideration all the interaction with other species or the environment. And also we don't consider the natural oscillation of the environment. So in the environment, for example, the organisms that they experience like high fluctuation of pH, temperature, oxygen, so we cannot control under laboratory conditions. So a way to get over these issues, we are trying to use this kind of system, natural knowledge, that are environments that have similar characteristics as the future environments of higher temperature, lower pH, sometimes increased CO2, oxygen as well. So, and they are distinguished by shallow volcanic event sites, and these are a few examples. Vulcano Island, so in Sicily, Italy, where we've been working with the Two Frontiers projects, and also Shikinejima here in Japan, where I spent my postdoc position, and also in Ambito in Papua New Guinea, where it's a great place. And then we have semi-enclosed bays that I'm not going to talk about that deeply, but they are different. They are closed bay, so where there are difference in pH, like lower pH, high temperature, sometimes ice sedimentation, so that occur all together in the same site. So they are tools for studying the effect of ocean acidification and warming at the ecosystem levels. And also chronic exposure, so like we have species organisms that live there since many years, so we can say if they have kind of adaptation to such environment, and also select winners and potential kind of adaptation. So we built this project called ICONA, which is International CO2 Natural Analysis Network, where we are trying to create a kind of network between the different researchers from different parts of the world to try to have knowledge from the different natural analysis around the world. So, and it started with a collaboration among University of Palermo, University of Tsukuba, and ERD, which is a marine center based in French and also in Caledonia. So, what are these shallow volcanic vents? So, they are located in volcanic area dominated by CO2. So, the seawater is either interacting with many volatiles, aromatic volatiles like CO2, like hydrogen sulfide, and then they are emitted at vent sites. All along this circuit, of course, seawater reacts with many minerals, like many rocks, and losing some and taking up others. So that means they are very energetic ecosystem. They are very dynamic. So this is an example. So this is Vulcano Island in the Mediterranean Sea. So you see here, there is in the right side, there is the main degassing, where is it? Yeah. Main degassing vent site with different dots, and you see you see the bubbling from the seawater, from the seafloor, the numerous CO2 rocks and marine fumaroles. They are mainly composed of CO2, more than 95%. Darker gases are H2, methane, hydrogen sulfide, that are few concentration ranging from few ppm to few percentage. So they release a kind of 3.6 tons of CO2 in this day per day. So it made how much CO2 you have there. And of course, this creates a kind of gradient because the CO2 is spreading all around the Levante Bay, as you can see in the picture. And this creates this gradient, we have pH ranging from less than 6, close to the inside, to normal seawater pH, around 8.1. So we use this area as natural laboratories to better understand whether species, organisms, also the community, may be able to adapt or acclimate to the long term in this environment, such environmental condition. So, for example, we choose some site where the condition of the pH, of the CO2, was similar to the one expected for the end of century for the entire globe. So, this is a video from John Kowitz, who also used the drone, and just to show you the venting site, which is this one. Try to show you this one. So this creates this kind of gradient all along the coast. So this is another hydrothermal shallow CO2 vents in Japan. So where we finally are going next week for sampling. And here you see also difference, in the side, you have you see the number there are the concentration of CO2. So you see the red spot are where there is high concentration of CO2. So the pH should be also lower than 7.7, coming from also 7.6, and also the blue side is the control side used for reference condition. And this is another shallow vents, is in Papua New Guinea, where I've been. It's very beautiful, I can say, beautiful place. And also Iwotorishima in Okinawa, where is it also I've never been there, but there is people working on this project called Econa, which are working and doing some research here. But what we found there, what we can say so far about this environment, what about the biodiversity there? So what we can say that in the area close to the inside, we have a loss of taxonomic and functional diversity. As you can see, on the right side, you see from the reference side to the MetaSUB side, you have a switch from shift from complex ecosystem with other forming species, grasses, corals, and to a very simplified algae-dominated habitats, as you can see here. And this was demonstrated like in different environments, like natural analogs in subtropical and tropical ecosystems as well. So, but despite there was this like effect on the community, reduction in complexity and stuff, there is organisms that live there that should have some functional trait potentially conferring capacity of adaptation to such environments. So we've been studying like sea grasses, fish, corals, polychaetes, gastrophods, not only me, but many with other many researchers. So for sea grasses, for example, you see some experiment we did in Vulcano. This is Ischia, and close to Naples. And you see that they thrive very well under isotope conditions because they have a strong affinity for dissolved organic carbon, such as CO2 or bicarbonate, that they use for like they can stimulate primary production, so they increase like photosynthetic rate, also stimulate crops. So they thrive and they can live very well under such environments. But for example, corals, what we can say is there is a reduction in complexity when you look like along the gradients. So there is more pH, there is less complexity. So there is a shift from the corals, complex coral, branching corals, to very simple corals and tarfy algae. So there is also, as you can see, there is reduction in complexity, with increased CO2. And also here for coral classification. So there is some species that like reduce the calcification rate under isotope condition close to the vent site. Other species that are like, they have similar responses. So this means that there are winning species and losing species. So winning species that can live in both the sites and control in isotope sites, other species that we can find only in the control site, in the normal CO2 condition. So, but for other benthic animals, for example, this gastelot we are studying now, this is like, we found two species in close to the vent site that are smaller than those found like in normal pH condition, and higher mass specific energy consumption, and in general, a lower wall animal metabolic energy demand. So it means that they have kind of trade-off between the activities that allow these species to maintain calcification and the repair like the shared solution. But also for, I'm not going deep into these other organisms, but we found like physiological and also genetic adaptation to such environments, with population that were different between the control and the vent site. And also I've been working on fish during my PhD in Cano Islands, and we started to understand to assess whether they have like kind of difference in the behavior. So we studied the behavior of these two fish species that are chronically exposed to the isotope CO2 condition close to the vent site, and try to understand what happens for like their behavior. So we found difference in the behavior in terms of like adaptation, in kind of anti-predator responses, in mating behavior. And so what's next? So there is also a huge, highly diverse microbial communities at the vent site with a large variety of metabolic strategies. I'm not a microbiologist, but I've been listening to many microbiologists so far. So there is a lot of like these electron donors from the vent site, so that are used as electron donors by chemically prokaryotes, so to obtain energy and fixing CO2 like for producing my biomass like for the entire web, the trophic web. Electron absorbs like beyond oxygen, and so this means that these systems are very dynamic. They offer like sometimes unique biodiversity. So unique microorganisms as we, well, maybe I lost one. Okay. Yeah, this one. So that's why we went there with the two frontiers. We collect samples, sediment sample and water sample. They discovered this channel bacteria was able to like a very efficient way to sequestrate carbonates. So, but I'm not going to talk about that because I think already Brendan talked and spoken about that. So, but what this area can also, this natural environment, natural CO2 environment can also provide an important natural contribution because there is like acid evolution, acid gene flow. These are new approaches, are not very well studied so far for this ecosystem. So nature-based solution of adapted species that could represent a source of like resilience, gene of resilience for that may be like transferred to ecosystems that are threatened by climate change. So potential mitigate and counteract the effect of loss in biodiversity due to climate change. Also discovering organisms capable of controlling CO2, as we I showed before. And also fuel surrounding ecosystem, stimulate carbon fixation and export by primary producers. So they can be a source of energy for adjacent area and nature-based solution. But despite that, we started to like study these volcanic active vents and SIP in the entire globe, and we discovered like that more over than 200 vents and SIP around the world. Most of them are like volcanic geothermal vents. Then there are top marks, there are hydrocarbon seeps, mud volcanoes. But this environment are treated, are shallow ecosystems, so less than 200 meters, are treated by anthropogenic activities. So trawling, bottom trawling, seabed mining, and other activities. So that the problem is that only few of them, very few of them are highly protected. Most of them are like, and these are that are protected are in remote areas with lower anthropogenic pressure. So that means and that highlight that it's essential to emphasize the importance of expanding protection to this environment, even because especially if global CO2 emission continue to rise at this level. So thank you for listening today. I'm very glad. Thank you. And also thank you very much.
Any questions?
Very selfish. First off, great talk. Thank you very much. Very selfishly, I'm curious. We talked a little bit about sort of, other interesting events that would be worth sampling. So to have been you and your team, we've gone volcano, we've sampled those 7-meter seeps, going to Shikinojima. Where in the world do you think it's worth looking for another sort of biologically divergent site, like these seeps where you would expect, a very different microbial physiology than from what we've seen before?
What do you mean, like?
So it's like the example I can give is volcano. Those were warm seeps, it was warm water coming out. Those ones at 70 meters, the water was like 17 C, right? So it was pretty cool. And I know Papua New Guinea has different stuff going on. I'm curious, like from a geochemistry standpoint, where should we look? Where should we expect to find stuff that's completely different?
I think it's hard to find similar things among the different natural animals and different plants because they are sometimes sampled in tropical environment, sometimes in subtropical or temperate. So they have different things, but what I think the most important goal for us is to see whether they can help understand how the species organisms can also try to adapt to this environmental change, just like a sort of window to the future. But I think it's not very easy to do that because, of course, these are only prediction. But of course, there's species that are more tolerant, so probably we expected maybe more tolerant to climate change, other species that are less tolerant. So I think it's the most important things when studying this kind of ecosystem.
Yeah, following up on that question, how many total sites around the world do you think could be good carbon sinks? Like if we could, because if there's hundreds of thousands, maybe say there's only 5,000 sites, I'm trying to get something from every single one of them and have a genetic catalog of all of them. I mean, I don't know how many there are, I'm just curious.
Yeah, I think so far we only discovered a few of them. There are many around the world, but sometimes we use shallow because it's easy to like sample there because there is, of course, shallow volcanic vents are more than 200 meters, so only a few people can go working there. But so far we discovered many, and I'm not talking only volcanic vents, but there is pock marks, there is mud volcanoes, yes, that are also very unique environment. But I'm focused on that because I didn't have much time to talk about other stuff.
And some of the mud does also often have sulfur, which is a big part of it. Yes. Some of them see the different co-factors, but they're very smelly as well.
Yeah.
But if you can find out how many, then we should just get all of them.
I actually have a question over here. Hi. So, not to minimize the importance of carbon cycle and whatnot, but I'm curious about the nitrogen cycle. I feel like it's often overlooked in nitrogen fixtures and the bioavailability of it. Have you looked into any areas of the nitrogen cycle within these extreme environments that could be of interest?
Not yet, but it would be nice to have a look on this kind of environment, of course. But yeah, my point of view is more like ecologist, like more marine ecologist, working on big stuff. So I leave this job to other researchers, which are at most more skilled than me, of course. Thank you for the question.
All right, I think in the interest of time, we're gonna continue with the next one. Thank you so much. Right, for the last talk of the session, we have Gabby Carpenter. Gabby is a PhD candidate at Louisiana State University. She studied the ecology of mesophilic coral ecosystems and coral disease. Her research focuses on coral disease at multiple scales to support marine protected areas.
Hi, everyone. Thank you so much for having me here today. I'm going to maybe get even bigger with our scale here and talk about coral disease and examining potential reservoirs for pathogens. And I hope that you can kind of find some parallels in your work, even though we're not talking about metagenomics here. We're still looking at spatial scales and how things are connected. So first, Brayden gave a nice overview for us of corals and the whole biome. I'm going to do that quickly again. So we have this complex animal that hosts the symbiotic algae. And on top of that, we have all the bacteria, everything else that goes into this organism. And that makes studying them and being able to identify pathogens very difficult. This is one of my favorite photos of coral polyp. You can really see there the symbiotic algae, and I just think it's such a beautiful photo. So then when we scale it up and look at marine diseases, there are many causative agents that could infect corals. And it's really hard to identify. I think the fact is there is something like around 40 identified coral diseases, and we have identified 2, maybe 3 actual pathogens that infect. And some of those have been debated. For example, white plague, we thought we had a pathogen, and then we kind of decided that we actually weren't able to transmit, and it's really hard to identify these, which makes studying coral disease really difficult. On top of that, you add in the element of corals interacting with the environment, and you bring a whole other thing in there. So that's kind of where my dissertation starts to come in. And then also when we look at marine disease transmission, we have so many modes of transmission. So you can look at ocean currents, which is what I'll be focusing on today. This kind of widespread ability of potential pathogens to reach reefs. Ballast water is a new thing. Stony coral tissue loss disease was a disease in the Caribbean that appeared in 2014. It started in the port of Miami, and there are a few theories that, and some confirmations that actually ballast water was able to transfer this disease from the port of Miami to Caribbean islands, and then start an infection at an island. Sediment is another great example. It can often serve as a reservoir and a mode of transmission for corals on reefs. And then we have marine organisms. These three very colorful organisms here. We have parrotfish, fireworms, and drupella. These are all corallivores, so they are eating corals and moving on to the next one, eating another coral, munching on it, and this could also be a way to transmit disease once it's already on the reef. So for me, I am trying to really understand what some of these ways that we can transmit disease, and to do this, I'm looking at multiple scales. So I'm going to step back and kind of put this presentation in the larger context of my dissertation. My first chapter is kind of looking at molecular mechanisms of disease, specifically that interaction of corals in their environment. I have done a transmission experiment under different light conditions, kind of acclimated corals to different light and transmitted disease, taking DNA samples, RNA samples, sediment samples, and haven't really analyzed much of that yet, so we'll be able to talk about that today. But just kind of seeing how you could potentially change transmission with environment. Next, scaling up, I'm looking at a community level of disease transmission. We've taken 3D photo mosaics across time in my study site, Flower Garden Banks, and I'm working on building a simulation model to see how disease transmits within a single reef area across multiple patches. I also wish this was further along because seeing some of these models and this imagery is really, really cool. It's really fun to kind of go underwater with the camera and marry that kind of photography and science lens, and come back and be able to build these models and show people what you're able to see underwater as a scientist. But then finally, what I'm going to really be talking about today is kind of taking it even bigger and looking at the larger seascape. So I'm studying in the Gulf of Mexico and trying to understand how these isolated reefs there have been infected with disease, and looking at potential upstream sources. So as I mentioned, I work in the Gulf of Mexico with proximity to Louisiana, specifically at Flower Garden Banks National Marine Sanctuary. So this is a NOAA sanctuary. It has a NOAA sanctuary office with lots of scientists doing amazing research there. It's a very unique system. You can see in this photo here, it's very isolated. Those far northern red dots are the sanctuary itself. And when you look at how close it is to other reefs, it's anywhere from about 500 miles is the closest reef in the northern Yucatan Peninsula. And then, the Florida Keys are off over 700 miles away. Despite its isolation, it has an incredible coral community with over 50% coral cover, which if you're thinking in the Caribbean, that's absolutely unheard of today. And the colonies that are out there are massive. We're talking about the size of small cars. So they're hundreds of years old, and they're somehow really thriving in this isolated environment. And historically, there's been really low disease incidence in this area. So with climate change and more recently, more connected the world is, we've seen an increase in marine diseases and coral diseases. But here, there's almost been only no other outbreaks. We had a disease event in about 2005, but otherwise the reef has kind of been thriving and seems to be resistant to some of this. So to get a little visual, I have some nice photos. Here's some of the amazing corals that I was talking about. This Orcacella colony on the left is just huge, and you just get this everywhere. So in August of 2022, there was a coral disease that broke out that was unknown. At first, we were all very nervous that it was stony coral tissue loss disease, which is absolutely rapidly moving and has kind of just killed reefs really quickly. We believe now it's a white plague outbreak. But these are some photos of what coral disease looks like there. Coral disease is also difficult to identify in the field with the eye because you have the same stress response that you see all the time in corals, which kind of looks like bleaching. There are a few ways in these kind of like white plague forms to look at what it actually is, whether or not you see these lesions on the edge of colonies, if they're starting in the middle, what kind of tissue loss you see, is the tissue like sloughing off, can you see it around you? But overall, identifying these in the field can be really difficult too. And then kind of to orient you to all the oceanography that will eventually be very important for this talk. So here we have the Gulf of Mexico. And the main current running up through here is the loop current. So this brings warm water from Central America area up towards Florida and eventually joins the Gulf Stream. So this is the primary current working in this area, and the loop current is very unique in that it tends to change. It has a lot of seasonal variability, so sometimes the loop current extends up into the Gulf of Mexico like this, and at times it can get all the way towards the coast of Louisiana and Texas, while at other times of the year, it goes more on a direct path to Florida. It's very hard to predict this. A lot of people have been trying to model and figure out exactly what conditions might cause this current to do this, but it's largely unknown and difficult to predict. And then what happens once you have this kind of large retraction and extending of the loop current is it eventually breaks off and forms these eddies. And so these eddies start moving and they start propagating westward in the Gulf of Mexico. And this is what we think is potentially causing a lot of connectivity and movement of pathogens and whatever else. This can also bring a lot of warm water over towards the reef areas that I'm interested in. And then the last thing you have is just kind of these northern and southern currents that go along the western Gulf of Mexico. And this can connect, Flower Garden Banks to some of the other reefs that are on that northern side of Mexico. So before I get too much into the coral, I want to convince you a little bit that these diseases can actually make it this far. So I want to look at diadema or sea urchins as a case for coral diseases traveling these kinds of distances. So as early as 1983, we've had these really bad cases of sea urchin die-off. And this is actually really important for reefs. Urchins eat algae and tend to manage a lot of the ecosystem on reefs and help allow corals to thrive. And when urchins die from disease or anything else, you see just algae growth start to take over and it can kill corals. So it's still important to corals, but, and so this is actually a paper from 1984 that just looked at chlorophyll content in satellite images to see if the disease that was happening and spreading could actually have made it from down in Panama up to the Gulf of Mexico. So these are just some dates being shown, but really all I want to highlight here is that pink arrow, where they were able to kind of track between the satellite images and the dates and say that this kind of path and progression of diadema disease definitely could have been through ocean currents. Another paper in 2006, this one used gliders, so a little bit more up in the technology, and they were able to show that this connection between the Yucatan with the reefs there all the way up to Flower Garden Banks does exist, and it's on the order of like 45 to 55 days, depending on exactly where you're coming from. And you can also see there's kind of that loop, that pink line loop is another way that currents can travel through that area. So now that we're there, I'll get into kind of the methods of what I'm doing. So I'm actually using biophysical modeling to kind of look at these connections and these time zones. And this is a Lagrangian particle tracking software, the connectivity modeling system. So this combines both the biological and the physical. Because we really don't know much about coral diseases, the pathogens that are potentially at play here, we're just modeling passive particles. So we haven't really attributed many biological characteristics. For some other work that people are doing in my lab, they can model vertical movement, swimming behavior, everything like that in this software. But for this study, we're just looking at passive particles. And then we're using hydrodynamics from the Gulf of Mexico HICOM. This is a 4-kilometer resolution grid that we use to use the forcings and look at the particles. So the biggest thing here is I just talked a lot about these currents and how the loop current moves from Central America up to the Strait of Florida. But now I'm going to maybe confuse you a bit and let you know that when we run this model, we actually run it in reverse. So we run this all backwards because we want to start with the source that we know, which is Flower Garden Banks in the north, and see what the potential connections are. So just to really highlight this, I'm going to show you the same images on the title slide running in reverse. So you can see the Gulf Stream is coming south. The loop current is running the other way, going towards Mexico. And that's how we run this model. So we release particles from Flower Gardens National Marine Sanctuary. And we've released a total of 200,000 particles per year. We do this seasonally, so 4 times a year we're releasing 50,000 particles. We release across depth from 0 to 45 meters to capture the entire reef area. And we release those two banks there, east and west, so releasing both of them. But when we total all of that up, it's 200,000 particles. And then we also wanted to look more closely at the known disease event in August of 2022. So there we did weekly releases of particles 90 days before the disease event, just to give a little bit of a finer picture and see what was happening close to that time. And over those 90 days, we released a total of 280,000 particles. The number of particles that you release is a little bit arbitrary. It's mainly about kind of saturating all the connections that are possible without it being too computationally intensive. So there's no real equation to figure that out. It's just kind of a little bit of a trial and error, but this tends to be a good sweet spot of we saw all the connections that were made without it taking too long. All right, so for some of our results, first looking at those annual releases. So we broke up some of the results into generally higher loop current intrusion years, so when the loop current is making its way closer up to Texas and Louisiana, versus lower loop current intrusion years, where we tend to see that more direct path to Florida. And a bit surprising, in these high loop current intrusion years, we actually see that the particles are kind of staying in the Gulf of Mexico. So to orient a little bit more to the graph, we have this color scale where these cooler colors mean that there is not as much the dark colors, whereas the yellow means that more particles reach that area, and this is a probability density function. So we're looking at on a log scale. I have highlighted all of the coral reef areas in the region in pink. And then we have these non-convex holes looking at 15 days from the release, 30 days from the release, and 60 days from the release. So we can see closest to the sanctuary, the dotted pink line is where particles were about 15 days from release, the dashed line where they are 30 days from release, and then the solid line is 60. And you see that we have a lot of particles still kind of up in the northern Gulf of Mexico. They haven't really made their way to any of those coral reef areas in the Yucatan Peninsula, although they've kind of started to reach some that are on the northern part of Mexico. If we look at the low loop current intrusion year, we actually see quite a big difference here. We see that within 15 days, we're still kind of close to the sanctuary up in the northern part of Mexico. But once we get towards 30 days, we have really started to reach out into the Gulf. And by the time you're at 60 days, you have pretty much saturated the entire area, and connections have been made from Flower Garden banks all the way to the other reefs. So that was pretty surprising. And it sounds a bit counterintuitive that when the loop current is high in that area, it wouldn't connect as much. But what kind of happens with the loop current is that when you have this high current, it tends to divide the Gulf of Mexico and actually almost keep particles or pathogens or whatever it may be in the water kind of north and separated. Whereas when you have this low loop current, it allows for more connectivity and those eddies to kind of start to play a role, we think, in transferring all the particles around. So that's why we kind of see this large saturation of the Gulf, despite the current maybe moving more directly towards Florida. So now I want to just replace that 2018 year with our disease year, 2022. We can see a pretty similar pattern to another lower loop current intrusion year, 2020. But almost more notably is this connection to the Yucatan. So let's see if I can. Okay, I don't know how it's working. But anyways, if you look at that 30-day non-convex hole, we can see that kind of lighter blue that's going right by those reefs around Cozumel in that area of Mexico, which are known to have diseases. So that's an interesting result there, and looking at the year where the disease appeared, it does seem that some of those connections to reefs upstream are much stronger, even than in other low loop current intrusion years. And then just to show a nice little pretty video, I have here looking at the weekly releases of the disease year. And this is just highlighting a little bit more visual of this kind of going back. So here we have 15 days and where all the particles were. And then as we keep spreading, we have the 30 days, and then there we start to make that really strong connection over to Cozumel and the 60 days. Just kind of a nice visual, seeing it all spread out, and also really drives home that idea of backtracking from the area that we know to potential upstream sources. All right, and then lastly, just another way to look at these results. I've showed you a lot of fun maps, which are very pretty, but the other thing we can do is look at just the overall dispersal distance of the particles. So these are the results from the annual releases from 2015 to 2022. And what we've done here is use a violin plot of all of those particles. So each one of these violin plots is made up of the 200,000 points, and it's the maximum distance that the particle went to. So we can start to see some patterns. This is newer data that I'm just starting to tease apart, so I haven't gone too much into this. I highlighted them both by season here as just kind of a way to look at, are there certain times of the year where particles tend to travel further, thinking about if that aligns with certain temperature conditions, or some diseases tend to have a seasonal, or some diseases have kind of a seasonal component to when they tend to arrive on reefs or when they tend to be worse. So we can see kind of a general trend actually from 2015 to 2022 of just higher connection. These last few years seem like particles were traveling much further than they had before, which is an interesting result. I'm working right now to increase this data set. So I'm hopefully going to go all the way back to the year 2000 so that I can add in that other disease year of 2005 and see if we saw similar currents in the region during previous disease outbreak. And also hopefully kind of add to this data set here of looking at dispersal distance and seeing if we see that similar trend of a rise in dispersal distance and just a greater connectivity, or if there's more of a seasonal component to these kind of things. So that's where I'm moving with this, but wanted to show just the initial figure now. So lastly, what are some of the implications of this? Why are we doing it? Well, we see that connectivity matters. There are lots of things constantly moving in this environment, pathogens potentially being moved from reef to reef across this large scale. And the hope is that this is going to help inform the sanctuary. If we are able to see these previous patterns of connectivity in times where the loop current might make a difference of exchange, how can we use this to potentially build models for the future and be able to project a little bit, or just know if we have a perfect storm of bad disease outbreak in Mexico and currents kind of doing this thing now, can we be prepared? Can we potentially use coral probiotics as a mitigation in advance of disease arrival to know that something may be coming? And then lastly, to add climate change into this, the loop current is expected to change in the future because of climate. It's actually expected to decrease. This is a figure from a paper that modeled a decrease in the loop current under future conditions. And so while initially this may sound good or sound bad because the low loop current intrusion years have this high connectivity and this saturation of all these upstream sources and reefs, it may actually be a good thing because part of the reason for that connectivity were these large eddies that come off of the loop current. So if the current itself is moving a bit more directly to Florida, we may not see those kind of spin-offs and those large eddies that tend to propagate west and really fill the Gulf. And this also, because the loop current brings a lot of warm water from the Caribbean, we may actually see decreases in temperature, which overall for corals can be good. And we won't see these higher temperatures that the loop current tends to bring to those reefs. So yeah, thank you very much. I hope you were able to find some parallels to anything that you work in, and I'll take any questions.
First of all, thank you for your talk. It's very interesting to hear why. I see a lot of this in my diving all over the world, so it's interesting to kind of put this behind it. In southern Oahu, we have an area where there's a lot of turtles that have significant tumors all over, and it seems to be some disease pathogen, but a lot of people I've talked to, they don't either seem to understand what it is. Some people say it's a herpes virus that's exacerbated by the nitrogen fertilizer that runs off in these well-to-do neighborhoods. Have you heard of anything like that or are familiar with that?
Not specifically in Hawaii, I haven't. I do know that in some of the areas we study in, like in the US Virgin Islands, there have been some ideas that this offload of like nitrogen and things reefs that are closer to the island may also increase disease. But as far as something like the turtles, I definitely don't work with anything that large. But that's interesting because you could probably like model this. There's a lot of higher resolution models if you're looking at coastlines, where you could really incorporate some finer bathymetry and stuff. So I wonder if you could kind of look at any potential spread of that. But I'm not super familiar with that now.
Thank you.
Yeah.
Thanks, Andy. This is going to be a little stretch, but the underlying data of these currents are what you're using for these modeling of particles. Could you take a similar approach using the airline connectivity between MetaSUB cities as the currents in the network, and then the people or the particles that you could model? And I wonder if some of the data that we saw earlier, each city has its own signature. There does seem to be some large, maybe not strictly geographical connections, these long distance connections that were pointed out in one of the talks yesterday. Maybe this is an approach that you could start to make sense of why those two cities look the same in machine learning models, because it's a lot of traveling directly or indirectly through it.
Yeah, I think it would definitely work. I mean, I think it would be require different software than what we're using, of course, but I think just having the idea of, movement, we're using ocean velocity here, but something else to the airlines and doing that kind of connection, I think it could work. I don't know exactly how to do it as a marine person, but I'm sure there's.
You know better than me.
I don't know, I think. Oh, sorry.
When the, great talk, by the way, it's fantastic. When the coral get infected or enter this disease state, is it pretty guaranteed to sweep through the population, or is it are there some variables that change the infectivity of the disease?
Yes, that's a very loaded question. There's a few layers here. I'm going to start first with it does depend on the disease. So I mentioned a few times stony coral tissue loss disease. That's probably one of the newest diseases we've seen. That tends to move through an individual colony and a reef very, very quickly. One of my lab mates is actually using this similar modeling to look at how that disease moves around an island, and it's quite remarkable how quickly it just moves from reef patch to reef patch. The other thing is, it can be very different, but also not all diseases infect all species. So we see different species susceptibility with all of these diseases too. So that makes a big difference too sometimes. And we're not totally sure why that is the case. If it's something to do with the symbionts, some people think the type of symbiont they host might make them more resistant to disease, all of those kind of things.
Yeah, just like hearing you talk about this, I can't help but think about SIR models. I don't know if you're familiar with those, but susceptible, affected, recovered. They used a lot for epidemiology, and I can't help but think about that with the coral. Really cool.
Yeah, so actually the model that I just mentioned that my lab mate is doing, that's an SIR model, where we are doing that. That's kind of the basis of his, and he's trying to expand on the SIR model and make different susceptibility categories for the different species of coral that we see have kind of some that die much quicker and some that don't die quite as fast. So yeah, there's a lot of papers on it actually. I can show you some.
I have time for one more question. I was just wondering about the correlation between disease and hurricane activity, thinking that hurricanes may stir up a lot of the sediment, probably transfer fairly large distances as well as increase their flow and runoff into an area like that.
Yeah, definitely. There's some work on that. I'm not sure entirely. I haven't looked, especially in this study, at any disease or any hurricane years. But that's usually a big factor. In some ways, in the Gulf of Mexico, for flower gardens specifically, sometimes hurricanes can be a good thing because it tends to decrease the temperature of the water. just by mixing everything up. But I'm not sure specifically how it may impact disease, but I imagine definitely the sediment component could play a big role.
All right, thank you so much, Gabby, and thank you to all our speakers. That concludes this session. We are going to reconvene at 2 p.m. For the global surveillance and metatranscriptomics session. If you order a bento box, they should be there outside, or they're going to arrive in the next couple of minutes. Lastly, a big round of applause to all our speakers. See you in 2 hours.
It's hard because I don't want to prioritize myself passing over behind. Yeah, I want to have an analogy. I mean, of course, there's many people I have about so, right, exactly. It's a good start. Yeah. It's the gateway back there. Yeah.
Want to check it out?
So my question is like,
Yeah.
sample these libraries.
You are on.